So before I begin, I just want to give a very brief introduction about Mission Bio. So people ask us all the time, how are we different from other single cell companies? And really, we're the single cell DNA sequencing company. We launched the platform Tapestri in 2017. And really, this was the first commercially available high throughput platform to look at targeted DNA sequencing. In 2018, we enabled custom panels. And so in addition to our hematology and our solid tumor panel, the ability to go custom allows investigators to look at any DNA that they would like. So any SNV or indel. So this really opened up the field to not only hematology and solid tumor researchers, but really anyone who's studying DNA. And then last year, we worked really hard to not only have SNV and indel, but to really open up those capabilities. And so we realized by just having an additional bioinformatics step at the end, we could unlock copy number variation, which we did. And then at the end of the last year, we also unleashed DNA plus protein from the same cell. So using oligo-tagged antibodies, you can tag the protein expression in single cells and then get information about SNVs, CMVs, and protein, all from the same cell, all across thousands of cells. So we're really excited to be known as the single cell DNA company, and we're even more excited to share with you today all of our publications that our researchers and collaborators are putting out there. It's really an exciting time for this new technology. So a couple of weeks ago, we did a publications roundup part 1, in which we really covered kind of cellular atlases that are being created because this is a new technology. And so, you know, what we're looking at on the bottom right is a graph from Dr. Ross Levine's lab. And each column represents a single cell and each row represents a single nucleotide variant. And so we're starting to uncover in hundreds of patients with AML or other diseases at the single cell level, what mutations they have and which mutations co-occur with each other. So this has been really great to really see the technology thrive out there and start giving us an indication of what's happening in AML and multiple myeloma and CLL and solid tumors and so forth. And then we also have 2 preprints in which we show the capability of doing DNA plus protein simultaneously from the same cell. So that was a couple weeks ago in our publication roundup part 1. And today I will go into our publication roundup part 2, which is really showcasing how investigators are using this technology in the clinic and as an add-on to their clinical trials. So I'll first talk about this study using solid tumor for colon cancer. And then we'll talk about these 3 studies where it was used in the hematology space. And finally, we'll end with some pontification and thoughts about how the single cell DNA sequencing technology can be used for MRD detection. Okay. Just as a reminder, I'd like to highlight why single cell DNA is important and what important information you can gain that you wouldn't get from bulk sequencing. So single cell allows you to understand the zygosity at each SNV location, so whether it's a heterozygous or a homozygous. And this can have very important clinical applications like it does in MDS for the JAK2 locus. A big one is identifying co-occurrence. So whether multiple mutations are occurring from the same cell or in different cells. And then being able to detect at single cell resolution allows you to have great sensitivity to find rare cell populations. So going down to 0.1% with the platform. And all of this information from a single cell level allows you to reconstruct the clonality. So which groups of cells have similar co-occurrence or zygosity, And then with this clonality information, you can reconstruct phylogenetic trees of how these mutations were acquired and really start to understand how this clonality evolves during treatment, treatment resistance, treatment relapse. So again, our biggest applications are in hematology and solid tumor, but really this ability is also enabling investigators that are doing genome editing and mouse models. All right, so let's get into it. Clinical trials. So this paper that came out of Memorial Sloan Kettering came out last year by Rona Yeager and Jorge Ruiz-Filho. And they were interested in patients that were on MEK inhibitors. So they had cohorts of patients that had colon cancer. The major mutation was a BRAF mutation. And so these patients were on an EGFR antibody therapy, but some of them were resistant. So some of them didn't respond to therapy. And so they were put on a MEK inhibitor. And even then, some of them still didn't respond to therapy. So they wanted to answer the question, well, what are the resistance mutations that are underlying this resistance? And the major finding of this paper was that they found a novel MEK1 mutation, V211D, which had never been reported before. And so they looked at a database of Memorial Sloan Kettering patients as well as ATGC database, and out of 30,000 samples, not one of them reported this novel mutation. So really this is very exciting that they're reporting it for the first time. And so they wanted to use the Tapestri platform to look at single cell and figure out, is this novel MEK1 mutation co-occurring in the same cells as that BRAF mutation, or is it in different cells? And then furthermore, what is the zygosity of this very novel mutation? So they turned to single cell DNA sequencing and the Tapestri platform. I think the beauty, so the figure on top shows the timeline of the different treatments that this patient underwent. And then on the bottom shows when they sampled the patient. And so this was a frozen tissue sample. And I think the beauty of the Tapestri platform is that you literally can go back into the freezer to old samples and look at them with this very new technology of single cell DNA sequencing. And you don't have to deal with degradation issues as much as you would with RNA, which is easily degraded. DNA is incredibly hardy, it's wrapped around histones. And so even if you can't isolate single cells from very old samples, the platform works really well with frozen nuclei. And again, that DNA is in the nucleus and it's very pristine. And we have a proprietary two-step process in order to access that DNA in order to look at these SNVs and CNVs. So they went to very old samples. They were able to get a tissue biopsy. And when they looked at it with, so they created a PDX mouse model. And when they looked at those cells via the Tapestri platform, they saw that this novel MEK1 V211D mutation did in fact co-occur with BRAF. The major clone is in blue and it was heterozygous for both variants, but they also found two minor clones which were both homozygous and heterozygous at these alleles. So again, single cell really uncovering, whether these mutations are in different cells or the same cells. And they go on in their paper to do really beautiful biochemistry in which they show that this mutation is in fact indeed resistant to the MEK inhibitor that the patient was on, but there are multiple MEK inhibitors out there and they propose using a different MEK inhibitor for any patient that has this particular mutation. And then finally, I really like in their discussion, they so why are all these mutations arising? This MEK1B211D mutation wasn't found at baseline. It was only found after therapy resistance. And so they hypothesized that it's possible that the chemotherapy is inducing DNA damage and creating mutations. And so even though that BRAF mutation was the driver mutation, and you can put patients on a BRAF therapy to target those cells, what happens is you might suppress that clone, but you're giving rise to other clones with other mutations. And so this ability to be able to detect those clones, quantify them, and possibly switch therapies when you think that patient is in remission is a very powerful idea that a lot of investigators are thinking about when using this technology. I think right now we're doing a lot of retrospective studies where we're going back to the freezer and seeing what mutations are there. But it would be fantastic to be in a clinical trial that's prospective, where you're monitoring these patients to look at the mutations that are there when you think they're in remission. Okay, so next we'll cover, we'll move over to hematology. And this was a clinical trial done with Alexander Pearl from UPenn and Kat Smith from UCSF. And in AML, a lot of patients have FLT3 mutations, and gilteritinib is a FLT3 inhibitor. And so they had a cohort of patients from a clinical trial that were given monotherapy of gilteritinib. And so, but some of them failed. So some of them either relapsed or didn't respond to the gilteritinib. So they wanted to understand what are the mutations underlying that resistance. And let's look at those samples at a single cell level. And so here is an example of one patient. On the left is the bulk sequencing data. And the beauty of these samples is that they were sequential. So they had 4 time points prior to treatment, on treatment 1, on treatment 2, and then after relapse. So you can see at these different time points, the FLT3 mutation goes down as assessed by bulk sequencing, which is what you would expect given the fact that they're on a FLT3 inhibitor therapy. But what happens is it actually gives rise to an NRAS mutation, as you can see on the graph to the left. And then IDH2 and SF3B1, which are important in AML, appear to not change during the course of treatment. But when we look at the clonality at the single cell level, what we see is that those mutations are still there and driving relapse. So the interesting part is that NRAS mutation is actually co-occurring in the same cells with FLT3, IDH2, and SF3B1, and that's shown in the pink clone. And you can actually see that clone at 3 cells at on-treatment time point 1 and at 89 cells at on-treatment time point 2. And again, I think the allure of this technology is if we were able to detect that clone during treatment when we think the patient's in remission, maybe you would be able to see that NRAS mutation was showing up, and you can put patients on a RAS therapy or a combination therapy with the FLT3 inhibitor. I think the second thing to really appreciate from the single cell studies is that even though with bulk, it seems like IDH2 and SF3B1 don't change, you actually see that they are going down if they're co-occurring with the FLT3 mutation, but clones that just have IDH2 and SF3B1 shown in green are actually expanding during this treatment. So again, really great resolution using single cell sequencing. The other way to look at these data instead of a bar graph is to look at a fish plot. And again, you can see that a quadruple clone and the double mutant clone really expand during relapse, which they think is causing the relapse. And then finally, they went on to profile many different patients, and they show them So the major finding in their paper was that secondary mutations arise in the RAS pathway. But these NRAS mutations occur at different times. So sometimes, like in the patient on the left, you can detect this NRAS clone at very low levels at pre-treatment. Sometimes, as shown in the patient in the middle, this clone is only detectable when treatment starts. And then for the patient on the right, sometimes NRAS clone is only detected at the very end during relapse. So just very interesting when these mutations and these clones arise. So that was about gilteritinib monotherapy. And I think there's a lot of interest in the AML space to do combination therapy, because we know that many different mutations contribute to AML disease. And so this was with Dr. Marina Konopleva from MD Anderson and Dr. Andrew Wei from Monash University. And they did a big clinical trial looking at Venetoclax combination therapy. Venetoclax is a BCL2 inhibitor, which inhibits, which targets apoptosis. And so the great thing is they had this big cohort of patients that were given combination therapy, and they were able to look at the ones that didn't respond and do some single cell experiments. And the reason why combination therapy is so important is, again, there are many different mutations driving AML, and so we want to be able to target all of them if we can. But secondarily, in this cohort, older patients oftentimes can't get chemotherapy, and so it's these small molecular inhibitors that are the first line of defense. And so really being able to use more than one could be very beneficial for this particular population. So they went back to the freezer after their clinical trial, and they wanted to see what are the resistant mutations that are coming up from patients on Venetoclax combination therapy. So what they found, they had 3 cohorts. So they had the ones that responded to the therapy. They had a cohort of patients that initially responded, but then relapsed, and they call that adaptive resistance. And then they have a cohort of patients that never responded. So that's primary resistance. And so in the first group that responded, they typically had an NPM1 or an IDH2 mutation. In the second group that responded but then relapsed, they typically had an NPM1 and an IDH1 mutation. And using single cell sequencing, they were able to show that that IDH1 and NPM1 mutation co-occur in the same cell. They co-occur with other mutations. So in the blue clone, it's co-occurring with a FLT3 ITD mutation. And that other co-occurring clones that have PTPN, while they're there at baseline, they're not the drivers of relapse. They actually go down with the therapy, and it's really that IDH1, NPM1, FLT3 ITD clone that's driving relapse. And then in that third group that had primary resistance, so never responded to the Venetoclax combination therapy, they're finding RAS mutations, which is actually really interesting because it supports Kathy Smith's and Alexander Pearl's data, in which, you know, their patient cohort originally had a FLT3 mutation, and the ones that became resistant targeted that RAS pathway. So in this clinical trial, they also found a very similar thing, that RAS mutations cropped up. But I think what's really interesting about this cohort is that they find different clones, and that the RAS mutations are always mutually exclusive. So they never occur in the same cell. They co-occur with EZH2, but 5 different clones targeting 5 different RAS mutations all occur in different cells and never together. So really cool biology that you can start to see using single cell sequencing. All right. And then a very recent paper that came out of Agios Pharmaceutical also did more experiments after a big clinical trial. So Agios, they have ivosidenib, which is an IDH1 inhibitor. And so their patients all had IDH1 mutations. Some of them responded to the therapy, some of them didn't. And so they want to understand what mutations are driving resistance and relapse in these patients. So they turned again to single cell technology to get more information from these samples. I really love these big overviews of the patient cohort. So they had over 174 patients. The severity of this disease increases from left to right, and then the mutations are shown from top to bottom. So the very top row is IDH1, and you can see that every patient has an IDH1 mutation, and that's because they're in a clinical trial for an IDH1 inhibitor. But what they really want to understand are what are the other mutations that are arising in these patients that are resistant. And the number one pathway is the RTK pathway. So again, you know, very similar to what the UCSF and UPenn group found and the Venetoclax paper found. It seems to be in AML, this theme that the RTK pathway is being targeted. And so specifically from this paper, they were also interested in mutations that arose in IDH2 because Agios Pharmaceuticals also developed an FDA-approved IDH2 inhibitor as well. But before we look at the cohort that have both IDH1 and IDH2 mutations, I just want to show that what's really important to uncover is the order of acquisition of these mutations. And you can do this using bulk sequence, and you can infer based on the VAF or the variant allele frequency. And so that's shown here. And so what they show are the genes in the bottom right in blue. They tend to have a VAF that's greater than IDH1. And so you can infer that those mutations came first, followed by IDH1. And then the genes in red, they infer that they came after that IDH1 mutation. And those are all targeting the RAS pathway. So again, just showing bulk sequencing data and how you can infer the order of acquisition for these mutations. But what's really nice with single cell sequencing data is that you know, because you can reconstruct the clonality. And so here they looked at 9 patients that with single cell sequencing. And again, these patients, now they're honing in that have an IDH1 and an IDH2 mutation. And what they show is that in the majority, which is shown on the left, the IDH2 mutation comes after the IDH1. And not only that, it's co-occurring in the same cell. So that IDH2 mutation is the green clone, co-occurs in that same IDH1, NPM1, FLT3 clone. They do find examples where IDH2 is independent in different cells, and that's shown in the middle and to the right. But the majority of the patients had both of those mutations from the same cell. And so I think it's just really interesting biology. There's an IDH1 mutation, you inhibit it with a drug, and then a mutation in IDH2 crops up. So it's a redundant pathway. And when you have information on a single cell level, you can begin to see, think about molecular mechanisms underlying the different pathways. So finally, I just want to end just with a little pontification of how we see single cell DNA sequencing being used in the future. And really, it's in the setting of minimal residual disease monitoring. And so this is really being championed by Dr. Ravi Majeti from Stanford. He just put out a paper this year in which he talks about how he feels that this technology can be used for MRD detection. He profiled over 300,000 cells. And again, looking at remission, the disease is gone, but really you can detect it with single cell DNA sequencing. And so here is just a model of how we see single cell DNA sequencing fitting. So at the onset, at baseline, you would do bulk NGS sequencing and single cell sequencing, and then the patients go on to therapy and hopefully go into remission. Well, we know during remission, some of those patients are going to relapse. And so if we can do serial samples during remission, we might be able to catch the patients that will relapse and not only catch them, but put them on a different targeted therapy. Dr. Majetti was one of our very first customers. So at the very beginning, he profiled one patient, and here they found a quadruple mutant clone that was driving relapse. We were able to detect this clone at remission at 6 cells. And I think the power of this technology, how can you go down to 0.1% and really trust that that's truly positive? Well, the chance of having 4 mutations are very low in the same cell, especially those same 4 mutations that you're seeing at relapse. And if you see a clone with those 4 mutations during remission, you know you feel confident that those mutations are really there, and you might be able to change the course of therapy based on those mutations. You know that they're in the same cell. So just a little teaser, but I think this is the hope how we would use this technology. I know others are thinking about using FACS or ddPCR for MRD detection, but I think really the power and the benefit of single cell DNA sequencing is the ability to look at hundreds of targets at the same time. Okay. So here he just profiles 3 different patients, and he shows again this low level. So sometimes the clone, which is shown in red on the left, you find it at baseline, and that's the one that's driving relapse and detectable at remission. Sometimes the clone in the middle in green, you don't detect at baseline, but it crops up during remission. And then on the right is just showing the ability to distinguish between chip clones and clones that are pathogenic. So what does this all mean? Well, this is starting to emerge from numerous publications now, where the number of clones is predictive of who's going to respond to therapy and who is not. And so on the left is from Dr. Majetti's most recent paper, where he shows that the higher the diversity in the clones, the lower the chance for relapse-free survival. In the middle is a recent paper by Dr. Takahashi from MD Anderson, showing the exact same thing. The more clones that you find at relapse, the worse the prognosis is. And then finally on the right is from Dr. Ross Levine, and from left to right, we're increasing in severity of disease. And as the severity of AML disease increases, the number of clones that we're seeing at a single cell level increases. So really tantalizing data to show, the power that you get from reconstructing the clonality underlying these samples, as opposed to just discovering what mutations are there. So with that, Mission Bio is the single cell DNA sequencing platform. We have all the assays, custom assays, the instruments, and then an analysis software for an end-to-end solution. So with that, I want to thank you. And if anyone has any questions, I will take them.